thank the organizers for inviting me to participate in this wonderful 13th meeting of the African Society of Human Genetics. I remember the early days of the organization, and the last meeting I went to was actually in Yaoundé, and I would never forget my experience getting to Yaoundé from Nigeria. Well, let's really congratulate the organizers and congratulate all of those who have worked to make this meeting happen. So when I was asked to talk about cancer genomics in Africa, I thought really I just needed to give you an overview of my understanding of breast cancer and why I to really go back to my alma mater, to go back to Nigeria to really see what I can learn, having left America, having left Africa more than actually nearly 40 years ago. So what do we understand about inherited susceptibility and where we are now? Because I actually really think that we're at the moment now where we're talking about precision oncology, precision medicine, and everyone is really excited that Africa will be the place to innovate in genetics, and that by understanding the root causes of these diseases, studying genetics in Africa, we can really improve the health of everybody. So why is this important? Well, I left Nigeria and I started my practice at the University of Chicago. And in a human genetics meeting, we should do pedigrees. And I hope this is projecting well on all of your laptops, wherever you are. But I started my faculty job in 1991. And this family is a Black family on the south side of Chicago. And they brought their genealogy to me with extensive 5-generation pedigree. And as you know, because of the slave trade, a lot of Black families actually try to see whether they can maintain their genealogy so they can tell the stories. And so this woman had breast cancer at age 34, and I gave her the standard of care drug, which is adriamycin containing. And then she brought every member of her family. At least 4 people will come to a visit with her because she was young and they were all worried about her. Anyway, I ascertained the family, and then they participate in the breast cancer linkage consortium meeting. And after BRCA1 was identified, she had BRCA1 mutation. And then we went on to test other family members. But unfortunately for my patient, she developed severe cardiomyopathy shortly after getting adriamycin. Then because of her low ejection fraction, even though we had recommended that she should get bilateral salpingo oophorectomy, she couldn't, and then developed ovarian cancer and died subsequently. So this woman, within 6 years, developed breast and ovarian cancer. And she basically transformed my career, because then I went and wanted to know, why is it that this Black woman developed breast cancer? And I thought about the face of breast cancer when I was in medical school. So, of course, at the time that we were beginning our work, there was no one doing breast cancer research in Africa. And so Dijon Ho, who was then my postdoc, went to see whether we can collaborate with my colleagues in Africa. And I went to Senegal, and I met the surgeons and the researchers there. And we said, we're really looking to do a survey of indigenous women to see who was getting triple negative breast cancer and who looked more like the patient that I was treating at the University of Chicago. Cut a long story short, and we've now published a lot. Other investigators in Africa have published a lot. But we know that when you look at genetics across Africa, it's not exactly the same. You can't talk about East African genetics, West African genetics, South African genetics without really appreciating the diversity across the African continent. So whenever I give my talk, I said, I'm doing this for all women, because at this moment, breast cancer is not one disease. We see rising mortality. And while when we published our paper in 2009, the average age of the young women who were getting breast cancer that we treated was 45. Under 50, we also see that the highest mortality, when you look at this map from GlobalCan, was from Nigeria. And so we then doubled down. We wanted to know, why is it that this country had the highest mortality from breast cancer when in fact the incidence is quite low? So we began to think about, it's not about black and white. It's really about understanding the diversity of genomes that's driving this cancer. And so when we started asking about the diversity of genomes, this is a paper that was published last year, really going back to the Atlantic slave trade and trying to understand why and what is the level of genetic admixture across the African diaspora. And so we understand that 10 million Africans were forced out of Africa, and depending on where they left and how they left Africa, they landed in the Americas, and then of course through the Caribbean. But this paper really illustrated the level of genetic admixture across the African diaspora. And the fact that it didn't matter whether you came from Senegambia, coastal West Africa, Nigeria, or Congolese. What matters is that modern day Black women who self-report as Black, or who we call African ancestry because of the level of genetic admixture, they can be Black and white, they can be different shades of Black. But one of the things that is really clear now, after the murder of George Floyd, is that there's been inequity in the way Blacks are treated across the diaspora. And in as much as Black women have the highest death rate from breast cancer and were understudied, I've really made it a passion of mine to really go and dig deep. And when you think about what the face of breast cancer is in Africa, when we started our work, it's women who were young, and because people were blaming them for delayed diagnosis and advanced stage, we actually learned that it's not about the women delaying diagnosis. It's that when they go to the community doctor, or even if they wanted their cancer diagnosed, there were no pathologists and there were no doctors to diagnose their cancer. So that delay was often put on the woman, but it's actually not the woman, because they're showing up, and we have published a lot of patients. But what happens is that they're showing up with a more aggressive breast cancer at a younger age, no screening program, no viable prevention strategies. And that was the state of affairs when we started our work, and it's still the state of affairs. No wonder we are now really talking about the inequities in biomedical research. And so we started really thinking, why is it that they get triple negative breast cancer? What are the reproductive lifestyle factors? What are the genetic risk factors? Because we wanted to build a risk prediction model and leapfrog so that we don't have to do one size fits all for African women across Africa. So that's what my colleague and I, Dijon Ho, have done in collaboration. And I'm going to just really skip quickly through the epidemiologic risk factors, because we have done that, and we have a risk prediction tool that we have out there. But we started in Ibadan, and then we went to Lagos, Yaoundé, and Kampala. So we can say that at least in modern women who are living in these urban centers, they have the tools now based on the work that we have done. Is it perfect? It is not perfect, but we keep working on it. And what we have done in phase 1 and phase 2 is to recruit more than 3,000, nearly 4,000 cases, and nearly 3,000 controls. And we've published a lot on this. And this is a paper that I thought was really important to do because we did not select by age or by family history, because majority of patients who we had in our study did not report a family history, whether it is that they didn't know it or it wasn't documented, but they were young and they had overrepresentation of ER negative and triple negative breast cancer. It's published in the Journal of JCO. The paper was published in collaboration with Mary Claire King, and we found that if you look at BRCA1, BRCA2, and PALB2, 12% of these women, we did, 1,000 cases and 1,000 controls, had these high-risk alleles, and it was consecutive cases. And when we looked to see, whether they were over 50 or under 50, we found that majority of these patients who were positive, of course, you could say they had family history and under 50, but majority, 65% and 17.5% of those who actually had mutation did not report a family history. And that's why Mary Claire King I have been emphasizing the fact that if we ascertain based on a strong family history, we're going to miss the majority of individuals who actually have inherited predisposition, and that what we should be doing is making sure everyone has a chance to have their risk assessed. And so we've been participating in a lot of consortia, especially the Breast Cancer Association consortia. I know in the US, the carrier study, actually we published in 2018, and carrier study published just this year in the New England Journal, of course. But what we found is that if you look at those cohort study, these women were on average 62 years old. So breast cancer faced in the majority of the world is older white women getting older and getting breast cancer. And so we ask everybody, go and get your mammogram when you turn 50. Well, if you look at African Americans, the average age in the African American case control cohort that was just published also was 54, at least 10 years older than what we're talking about with these Nigerian women, Cameroon women, and women in Brazil. So, of course, we know the population structure is different. Africa is younger, but Africa is moving. Women are getting into the workforce. And we can say the same thing about prostate cancer. But my talk is really about breast cancer genomes in Africa, and why we started with inherited susceptibility, and why these high-risk alleles are important across the African diaspora. We also went to Brazil, the only country where you can actually maybe not talk about black and white, but there's also racism and segregation. So in Bahia, we looked at women in Bahia, that little island in Brazil where you see a lot of very little admixture. And the women who are getting breast cancer in Brazil also young, like the African cohort, and they also had about the same frequency of mutations compared to what is in the general literature. So why is this important? It's important because in European ancestry group and in white women, looking at the Breast Cancer Association consumption of sequencing these genes in more than 113,000 women, we picked up the signal in 1,000. And what do we learn about penetrance in this BCAC cohort? We see that BRCA1 is the highest predictor of breast cancer risk, followed by BRCA2 and PALB2. And then there's a drop to the other genes. And then this is in comparison to the general population. If it's a true genetic model, we have to say that these women in Africa also have this very high risk. And in fact, they're going to develop breast cancer before the age of 40. No wonder in our study, the average age was 45, and the women presented with advanced disease, but because there's no screening. So without screening, how can you have your breast cancer diagnosed early? Then let me look at heritable factors, looking at SNPs. And of course, there are lots of discussion now about the case of the missing heritability. How much proportion of all the complex disorders that we have heritable risk factors? Are there rare variants? Are there epigenetic variations? Should we now be putting polygenic risk, or should we think about gene-gene interactions, gene-environment interactions? And if we're going to do this, we can't do one size fits all. And that's why it is really important to do cancer genomics research across the African diaspora. So we made an attempt. We did the root study, and we wanted to pull all the data for from root study in Nigeria with the African American Breast Cancer Consortium to do a GWAS. And guess what? We, because of course, poor sample size, we got one hit on 3Q, and we've published on that. And we put everything, we combined, and by really thinking about developing a GWAS, starting with a root study from Africa, and then going back to pool data with African Americans because of the admixture, we were actually able to get a better odds ratio with ER negative breast cancer because our cohort was enriched for ER negative disease. So we've done more work, and it turns out that in fact this SNP really is in the a region that is very close to a major modulatory factor, and that could potentially have epigenetic control of TNSF10 expression. And so we've done additional work to in our lab, and this paper is published. And what we wanted to do was to actually look at how TNSF10 regulates antiviral and anti-tumor immune responses. And it's a long story. I'm not going to tell it. But when you do CRISPR knock-in and knockout of this region on chromosome 3q, you actually see downstream factors that are important in immune responses. And so if these factors can be regulated by SNPs and it's epigenetically controlled, how might that contribute to breast cancer progression, and how might that even affect immune response? So I have continued now to work on cancer immunology, trying to understand what is the highest area of investigation in cancer. And in collaboration with Michaela Gak, we actually also ran into a pseudogene in the complex structure that is near BRCA1. Not only is this pseudogene very important, it's a long non-coding RNA, it also has differential expression in different molecular subtypes of breast cancer. And by comparing normal and tumor, we see that this pseudogene has differential expression. And we've been really working hard to understand why this BRCA1 pseudogene and link RNA might actually be involved in tumor inhibition. For those of you who are not molecular biologists, NF-kappa B and the TNF interferons factors are really important, right? We are talking about cytokine expression that modulates tumor microenvironment. So we sort of modeled it in the lab, and this paper is published in Cancer Research because some of these cytokines can inhibit virus replication. And in the experiment, we showed that these factors can also, compared wild type to knockout, totally inhibit tumor growth. So that's exciting for us. That is, starting with the genomic architecture of tumors in this Nigerian women, we're gaining insight into biology. And so let me end by talking about some of the work that we are now continuing to do with the African Ancestry Breast Cancer Consortium. This is not work that is easy to do, but finally I was able to get Adedokun, my postdoc from Nigeria, who's been very active in the African Society of Human Genetics, to be the lead author of this paper that was just recently published in Nature Communications. And I'm bragging about him because I met him as as a statistician at the University of Ibadan. He got his PhD, then he came and did a postdoc with us, and then he had his Nature Communications paper, and now he's working for Amgen because, of course, the value of the work that he did and his interest in not only just translating research across the African diaspora, but leading efforts to precision oncology, precision medicine, because of his work within the Nigerian breast cancer study. So that continues to really now allow us to begin to think about whether we can do a polygenic risk score in African populations using the pooled data, and he was successful in looking at additional SNP. Now we are trying to publish a paper on lifetime and 10-year risk by percentile of PRS in Black women using data from the Consortium of Genetic Risk Prediction of Breast Cancer in African Americans. And the reason why this is really important is that we feel that if you can actually continue to modify this, and this is why we're hoping that we can collaborate with researchers across Africa who are interested, pull our data, that we can set the parameter on who we want to put on study, how we want to screen them. But we believe that we can now identify 5% of women in the 99th percentile, or if we make a cutoff of 95 to 99 percentile or 90 to 95 percentile, that have, if you look at the age of 40, you can see these women, if we take a threshold of 0.02, who have a risk that is equivalent to that of BRCA1 or PALB2, and this risk starts right from the time that they're 25 and goes really significantly high. And so if we can identify them before the age of 40, might we be able to downstage breast cancer across the African continent? That's a possibility, and that's research that we're hoping that we can actually conduct and test as a result of this more than 2 decades of work that we have done in the African breast cancer studies, which started in Nigeria. So why is this important? Well, we have fancy mammograms, and everywhere in the US and the UK, we know that mammographic density, or young women going to get a mammogram, you're not going to find anything because the breast is very dense. And so and African women have maybe larger breasted. So even if you try to do a breast exam, you're not going to be able to palpate that cancer until it has become a large cancer that involves the lymph nodes. And then, of course, we blame the victim that they didn't show up. They showed up. So we know that even in Nigeria, in my practice, this woman showed up, but because we were able to get MRI, we were able to screen her every 6 months, and we picked up a very tiny cancer that would have become BRCA1 associated triple negative breast cancer within a very short period of time. So we've been doing MRI every 6 months, trying to see whether we can perfect the physics and get these women to be able to have a chance to have their cancer diagnosed. So if I were advising ministries of health in any of the African countries, I would say don't invest in large scale mammogram and population screening. Just figure out the centers of excellence where you can train people to use modern imaging procedure and then be able to find early cancers. But we can even do better than that. Maybe what we can do is do blood-based biomarker and leapfrog and find cancer early when we can treat it. And the treatment is sort of where we are going with precision medicine and precision oncology. Because the treatment, and I wrote this editorial in the New England Journal in 1996, very naive, not knowing that in fact it would take us this long to take the biology that was so prominent, so promising in terms of BRCA1 germline mutation. This is a very small cohort of 43 patients with BRCA1 mutation. If they got platinum agent, they did incredibly well and survived. And if they were wild type, they didn't do well. And that's what got our researchers to go and find PARP inhibitors and do the research to figure out how you can take advantage of DNA repair pathways and use that to develop new drugs. Now we have PAP inhibitors that are oral drugs that are now approved for treating triple-negative breast cancer. We need to do studies in Africa, and that's why we can't wait. So what are the biomarkers that we're going to use to find people with HRD or homologous recombination deficiency? This is paper that I'm most proud of, because we know that the genomic architecture, what we started off with HRD signature, and now we know that there's double base substitution, there's indel signature. When we compare Nigerian women, we had 97 whole genomes that we've sequenced in Nigeria, and we compare them to black in TSHG or white in TSHG. This paper, we have more Nigerians in a study for the first time, right? And that's because we went deep to say we should study Nigerians and then compare other people, not vice versa, where we're comparing Nigerians to what people had already written. And I hope that we'll get this paper published and we can take this now into a clinical trial. Why? Because we know what the HRD signature is. We know that, in fact, a lot of the things that people have talked as early drivers in the general population is not really they're not the drivers in Nigerian women. We found that GATA3 is actually a very important driver. It's mutually exclusive with P53, and one of the really amazing thing that we were able to do, we've been able to look at the life history of a breast cancer genome, looking at what are the copy number changes that first happens. When do you have whole genome duplication? When do you have amplification? When is the point of no return where these tumors now become so widely metastatic that we can't get control of them? And in as much as we have these early changes, we are really gung-ho that we can now use at least these genomic early changes as biomarkers that we can take into the clinic. So let me end by saying that we're in the era, and I listened to a little bit of the presentation in the other panel, and in the interest of full disclosure, I'm on the board of 54Gene, and there are many other companies now that are really trying to bring innovation into Africa. Even after this pandemic, I do cancer risk assessment. We shut down, yet we were able to do more genetic testing because we adopted telehealth. We now can look at actionable germline variants. We can target screening. We can target interventions, target prevention, whether it's nutrition, vaccines, exercise. And in precision oncology, we now can do accelerated biomarker. So let me leave you with this review by Sayahan, who is talking about the host genetic background is the driver, and they published how the host genetics impacts tumor microenvironment. We know that GWAS, we can look at GWAS of tolerance to cancer and autoimmunity, and there's a balance. And then we can look at rare variants and look at BRCA1 with beta-catenin, all of these variants that are important and may actually drive tumor trajectory. And then we must have accelerated biomarker-informed clinical trials. I'm proud that we've started some of those clinical trials in Nigeria, and I'm sure that other countries are beginning to think about it. But what I'm really excited about is that we're now in the era of data science, that we can use real-world data with artificial intelligence and deep learning tools to deliver the right drug for every cancer patient at the right time. And I think the group that's gathered here can do that. So I thank you, and we'll take some questions. But my colleagues, I call them, were diverse. This was the last retreat we were able to do on February 14, 2020, before we all shut down because of the pandemic. I'm hoping that after the pandemic, all of us will be able to come together again and really brainstorm about some of the innovation that we're going to bring to Africa. Thank you very much.